let's pick up our discussion of amino acids and their characteristics by beginning to think about how those amino acids go together to form proteins, that is, the functional workhorses of all biological systems. In this diagram, I'm showing you that proteins are polymers of amino acids, and we remember that an amino acid is comprised of the alpha carbon, which is connected to the alpha amino group and the alpha carboxylic acid, and as well as an alpha proton, and then some functional group or side chain, which can range from something as simple as a proton in the case of glycine to something much more complicated like tryptophan or what have you, remembering that there's 20 different flavors of side chain that we encounter regularly. So a polymer is just a long chain with a repeating unit, and in the case of proteins, the repeating unit is the amino acid linked together via the peptide bond. So here's our amino acid, second amino acid in our chain. The peptide bond is represented here. It's an amide bond comprising the carboxylic acid, or now it's a carbonyl, of the first amino acid connected to the amino group, the alpha amino group of the second amino acid. Now I've omitted the amino group from this in this diagram. So we see alpha carbon, carbonyl, peptide bond, alpha amino group, alpha carbon, alpha carbonyl, etc. And of course, what gives the polymer its unique characteristics, remembering that a given sequence is what defines the function of the protein, as we'll see again and again, that is the primary structure, the primary sequence, the R groups, the sequence of the R groups of the amino acids here. That, of course, giving us a much more complicated sequence, or rich, information-rich sequence than, say, DNA with its only 4 bases. Now, a couple of important ideas. First of all, the peptide bond is planar. Here I'm drawing 2 resonance structures of the peptide bond, and we see that this is the form that we might normally see, most frequently see, but this is another resonance form in which the amide bond itself is drawn as a double bond, and there's partial charges distributed throughout that. Normally we have more of a resonance structure, and Linus Pauling might say that we have 40% double bond character in that peptide bond. That has some really important consequences, the most important of which is that we generally think of the peptide bond as being planar. Now, there's some other interesting things. Since we've been talking about DNA as being a more stable information storage polymer than RNA, we can also see that the peptide bond does something great to our peptide, to our protein, stabilizing it against a hydrolysis, right? So the carbonyl carbon, because of the double bond character in the amide bond, is stabilized against nucleophilic attacks. Just a fun factoid there. But this planarity is worth coming back to, and again, this is something that we discuss at length when thinking about the Edison article that you've been assigned to read. That planarity is a simplification, right? So here I say the peptide bond is planar, más o menos. This is the key piece of the key figure representing quite a lot of data presented in that paper. And in the x-axis here we have omega, which is the angle of rotation of that peptide bond. And you'll notice that we're not looking at the full 360-degree rotation. We're only looking at a fairly narrow range of possible torsional angles of omega. And we see on one axis energy in kilocalories per mole, the energy of that torsion, and then the number of observations. So there's a lot going on here. Let me draw your attention first of all to the distinction between the number of observations, that is actual crystal structures of peptide bonds, versus these calculated energies. So these are from theoretical studies and computer modeling studies of peptide bond torsions. In that case, we have 2 basic kinds of data that are presented here. The histogram tells us the number of observations times 1,000 that different torsional angles of peptide bonds have been observed, and so we see that, in fact, we are planar and mostly trans, 180 degrees around that peptide bond. So it tells us that by and large we do restrict ourselves to planarity, but it's not strict, right? There's obviously deviations here, and we can see where those deviations come from. If we think about the energy that is required to introduce torsional strain on the peptide bond, and I'm not going to go into details here, but there are several different energy models represented by these different colored dots that give us the possibilities, and then we see Linus Pauling's expression for the stability of that that peptide bond to torsional strain, and that's represented by this line here. It's just this E goes as sine squared omega, which is that rotational angle of the peptide bond. So yes, we're planar most of the time, but according to some energy, and it's worthwhile thinking about what's happened in that paper. And this, again, is something that we're talking about a lot in class and that you're thinking about as you write. So I really think it's worth reviewing what the scientific method is and how that plays a part in our thinking about the peptide bond. We always begin with an observation in the scientific method, or in this version. And our observation is that, well, we've deduced what the structure of the peptide bond looks like. It's an amide structure, and so we have some ideas about what we think that means. The observation then leads to a hypothesis, and the hypothesis is that the peptide bond is in fact planar because of the nature of the amide bond, or what we think of amide bonds as looking like. Now, of course, once we have an hypothesis, we want to probe that hypothesis, right? We want to seek to support it or undermine it. And so an experiment or an experimental result is that the alpha helical structure, and we'll talk about what alpha helices are, but they're proteins, so they contain peptide bonds. They're determined to contain planar peptide bonds, and this is happening in various groups are working on this, but not until the 50s do we discover that in fact the peptide bond is planar for certain. And that, of course, that experiment goes back and reflects on our hypothesis, and we say that our hypothesis is generally validated, right? We observe that they're mostly planar. Then, of course, we know that experiments that have validated hypotheses lead us to the presentation of theories. And it's worth pointing out, of course, that in science, a theory means something different than it does to a politician, for example. A theory is something that is well supported by experimental observations. In this case, the theory is now that effectively all peptide bonds are planar, with this caveat that really we're restricted to planarity by some energy that's required to torque that bond. And of course, we can keep doing more experiments. We don't have to rely on 50-year-old or 60-year-old experimental data. We have a whole bunch of crystal structures, as indicated in the last slide, that reveal that peptide bonds are by and large planar. And of course, we can feed back into our theory from experiment. So this, I think, will help you again to think about the Edison paper and the sort of the claims that are made in that paper about what Linus Pauling understood about peptide bonds. Okay. Moving on to some more discussion of peptide bonds. Here's two cartoons of the peptide bond. Here's the peptide bond itself, reminding you that we generally think of there being a directionality to this polymer, beginning with the amide end, right, the primary amine end, alpha amino group end of the chain, and heading in the direction of the alpha carbonyl. So here's our peptide bond, carbonyl carbon, nitrogen of the next amino acid, peptide bond between them, and we see that we can adopt a trans or cis configuration, and the trans configuration is strongly preferred, 4-to-1 ratio, strongly preferred over the cis, and it's obvious why, right? Steric hindrance here induces us to choose. We choose the energy minimum. We choose that one which allows us to avoid the steric clash, and we can even compute what that preference might be based on our observations of how often we see trans to cis, and then we can just plug in the numbers, right? We have an equilibrium constant here effectively that tells us that it's worth about a kilocalorie or so. Now, there's other torsional bonds that we consider frequently, and let's just give them some definitions here. Here again is our peptide bond. Here's one amino acid. There's the peptide bond. There's the new amino acid. And we defined this bond between the alpha amino group and the alpha carbon as phi. And then we defined the alpha carbon to the alpha carbonyl as psi. And those are bond rotations or torsional angles. So the bottom line is that, yes, even though we never really rotate around the peptide bond in its trans configuration, the polypeptide has lots of degrees of freedom because these are more or less free to rotate. The other important thing to note is that phi and psi refer to the angles within a given amino acid, not between. So we see phi of n, and this here would be phi of n plus 1, for example. Here's another cartoon, and again reminding you that here's the planar peptide bond, but phi and psi define the rotatable bonds of the backbone, their structures. And so in aggregate, when we look over the whole protein at its series of phi and psi angles, those describe the overall structure of the protein without respect to all the side chains. Now, of course, the side chains are driving us to adopt one phi-psi angle pair over another, but if we just look at all of those over the whole protein, we get a sense of what the overall trajectory of the chain looks like in space. Okay, let's talk about an individual. This is a picture of Ramachandran. He's very important to us in terms, again, of modeling the behavior of proteins. Just a little historical background. He worked in Bragg's lab at Cambridge. That's the Cavendish Laboratory at Cambridge from 1947 to 1949. This is not at exactly the same time, but a little earlier than Watson and Crick worked in the same laboratory on the structure of DNA. Those of you that are really interested in Indian history, you'll note that Ramachandran is from India, and he's working in the laboratory around the same time as the independence of India from England. So politically, must have been quite a balancing act for him at that time. Just an interesting side note. This is a photograph of him by the chalkboard with a couple of structures of proteins. And so here's a proposed structure of an alpha helix. It's worth pointing out that he proposed a controversial and was later proved to be correct structure for the collagen triple helix. So in that case, we have a triple helix of protein that wound around each other, and we'll see another model of that later on. What we like to think of Ramachandran for, though, is actually defining what the available phi-psi space of proteins is. And let's think about what he did. So again, he's trying to understand what the structures of proteins can possibly be, and he's just making some simple assumptions. He assumes the peptide bond is planar, right? It's an oversimplification, but it's a pretty valid simplification. And then he just builds models, right? Now this is physical models. These aren't computer models. This is just pieces of metal and paper and plastic, sticking them together, and he's looking for places where phi-psi angles in which cause steric clash between the partners in the model. And then what he assumes is that we avoid steric clash, right? And that's a perfectly reasonable assumption. The way we think about it now is that the structures find some energy minimum to occupy, and they don't really go outside of that unless there's an input of energy from outside. So then we can ask, what part of phi-psi space is allowed and what parts are disallowed? That is to say, what phi-psi angles for a particular set of amino acids, say, are allowed and what are disallowed by steric clash. This allows us to make predictions, really based on very simple models, predictions of the true behavior of proteins. And of course, we can go back and assay our predictions for their validity experimentally. But what he did was make predictions, and they amount to what we now know as the Ramachandran plot. And the upshot of this slide, in which we see our first picture of a Ramachandran plot, is that steric clash restricts the conformational space available to proteins. So here's phi angles, all the possible phi angles for a given phi, for a given bond. Here's all the psi angles possible for a given bond. And in blue here, I've sort of given you a sense of what's most intensely populated or what's most frequently observed in proteins. And this really is a reflection of his model building. Without even respect to a single real protein structure, he's able to deduce that most of the time, phi-psi angles are going to be restricted to a relatively small region of conformational space. Here again is that peptide for you, reminding you that there's a psi bond, and, I think I got those backwards. Oh, crumbs. Phi and psi. Well, swap them around. Beg your pardon. In any case, what I want you to notice is that there are two main regions of space here, beta and alpha. And these original designations have stuck, and now we'll think of this as the right-handed alpha helical region of phi-psi space and the beta strand region of phi-psi space. This is a really big idea. It tells us that only a limited number of structures are allowed in proteins, and the others are disallowed just by virtue of steric clash between atoms in the chain. So that really simplifies how we think about protein structure. But again, I want to emphasize that this is a prediction Ramachandran made based on some simple models. But we can look at real data to make sense of these predictions. This happens to be data compiled by, not all collected by, but compiled by the Richardson lab at Duke. And each of these spots represents a phi-psi pair observed in 500 proteins that are not sequence-similar proteins. So this is a diverse population of proteins, and what we see is just exactly what Ramachandran predicted. There are regions that are allowed and regions that are more or less strictly disallowed. An interesting detail here is that this plot omits glycine, and it's obvious why you might omit glycine. Those of you that remember your amino acid structures will know that the side chain of glycine is a proton, not restricting the rotation of that bond by steric clash very dramatically, and so it's a much more flexible amino acid and can adopt a broader range of phi-psi angles. We also limit proline. We also omit proline from this chart, and the reason is that the phi bond of proline is severely restricted. We see that here. Obviously, the side chain forms a ring with the backbone itself, and there's omega, the peptide bond, again going to be planar, but now we're restricted to this around of rotation here. Another detail that I want to point out is that proline also has a hard time, or is kinetically restricted in terms of switching between the trans and cis forms of the peptide bond, and I'm just going to throw that out there, and we'll return to that at a later date. But we've referred to the cis-trans proline isomerization in previous lectures, so here's another diagram of that for you. But in any case, because the phi bond of proline is restricted in its rotational options, we limit our data here. So there's caveats, always caveats, but by and large, we see that the amino acids follow Ramachandran's model quite nicely. A couple more little details here. What value are Ramachandran plots? I want you to file this away for when we start talking about how we determine molecular structures later on. These are data that are gleaned from a database of protein structures. This is human myoglobin crystal structure, and we'll define what a crystal structure of a protein is later, but basically it's a set of three-dimensional coordinates that allow us to determine, among other things, what the phi-psi angles for every amino acid in the chain is, or for every for every amino acid in the chain. And so what we see, of course, is that most of the stuff falls into these allowed regions. Again, this is the beta region, and this is the alpha region, and we would say, without even looking at a model of the three-dimensional structure of myoglobin, that it's a helical protein. All of this in the right-handed alpha helical region of phi-psi space. And there's an outlier, and we don't know what the details of that outlier are, but there's a violation there, and what you'd probably guess is that there's some reason for that violation, structural reason for adopting that outlandish phi-psi configuration. Before we leave Ramachandran plots, again, I want to remind you that it's an extremely important idea, the ability for us to make the conceptual leap from simple model building to observed reality. Let's look at glycine. We see in myoglobin, that's again same structure of myoglobin. Most of the glycines are in our helical space, but we see some outliers that go outside of the allowed regions of phi-psi space. And then prolines too. Proline, again, as I said, restricted in terms of phi angles to about -60, and we see, in fact, that all four prolines, even though they adopt a wide range of psi angles, are restricted to that piece of phi space. And preprolines as well, and we won't go into that, but the residue just before prolines are also restricted, again, because of the ability of the difficulty of that cis-trans isomerization. Okay, we're coming to the end of this portion of the lecture. We haven't actually seen what a protein's secondary structure is, but I'll remind you that the primary structure of the protein is its amino acid sequence, the identity of the amino acid side chains. But I've given you the idea that just based on phi-psi preferences, there seems to be two regions of configurational space. We've named those alpha and beta. Again, that's 75-year-old terminology that sticks with us. Alpha helix, we'll see in the next lecture what an alpha helix is and what a beta strand is. The key to this slide, though, is that amino acids seem to adopt particular structures depending on steric hindrance. Some amino acids prefer to be in a region of configuration space, just like glycine is happy to avoid the defined regions of configurational space or phi space. Other amino acids prefer to be in one configurational state or another. So let's just take a quick look at these. These are all ranked. There's the 20 amino acids ranked by their degree of alpha helical propensity, and glutamic acid seems to have a high propensity for alpha helix. If we take a quick look, there's another kind of structure called a beta turn, which we'll define in a few moments, and that seems to have like an opposite tendency. So, and noticing that glycine, this will come back to haunt us, glycine has a very high propensity to find itself in a beta turn. The other factoid that I'm going to throw out here is that there are a couple of amino acids, isoleucine and valine, which are beta-branched amino acids, that seem to prefer to be in a beta strand configuration. So, again, another point to make clear here is that this is all statistical information, right? We can decide that glutamic acid prefers to be in an alpha helix based on thousands of protein structures that are available for us to study. What does that tell us? You get the sense now that what we can start to do is look at a primary sequence, a primary structure of a protein, and try to draw some conclusions about other elements of structure, secondary structure in this case. But what I want to make clear is that we can start to go even beyond secondary structure to tertiary structure, and from thence to function. The point being that starting very simply here, going from primary structure to predictions of secondary structure, we can also begin to look at the amino acid sequence and make decisions about what we think the fold of the protein is and what the function of the protein is. That's going to be an important set of ideas as we move forward. What can those predictions tell us? Here's a website that we're going to get familiar with over the course of the semester. It's called ExPASI, Expert Protein Analysis System. It's a set of online tools that allow us to do a variety of things with protein sequences as well as nucleic acid sequences, and one of which is to feed a primary sequence in and get out a secondary structure prediction. And this is an output of one of those pieces of software, and this happens to be, again, myoglobin. And what we see is that we can make a prediction just based on what we know about most protein sequences, that there are helical regions of the protein. Here's a couple of different prediction methods. Here's where I got the sequence information. These are important websites, of course: the National Center for Biotechnology Information, at the National Library of Medicine, at the National Institutes of Health, paid for by your tax dollars. So there is actually value to paying taxes, unlike what certain politicians might expect you to believe. The structure information, this is real structure information here from the structure, crystal structure, three-dimensional coordinates of myoglobin derived from the protein data bank, which we're going to discuss in subsequent lectures. For now, just accept that there's a database full of protein structures out there for us to mine, but we can use this protein secondary structure prediction algorithm, which you can access, again, via ExPASI, that looks pretty good, right? This application called Agadir tells us that the predicted helical regions of the protein more or less seem to match what we actually observe experimentally. All right, well, this brings us to the end of video 2.3. Tune in next time for where we look much more closely at the available secondary structural types for proteins. See you there.